I think we live in a golden era of science. I think we need to trust science. I think we need to have access to science.
Welcome to the CureGRN podcast. We exist to bring together families, doctors, and researchers to work together towards a cure. And now on with the show.
So precision medicine to me, both personally as a genetic counselor, is recognizing that each individual's genetic makeup and environmental exposures really plays a huge part in their health story. I think what we see a lot of times, something that's not well understood, is that typically when physicians are seeing patients, they're utilizing guidelines which broadly tell them how to approach patients. And these are guidelines that are made from large research studies focusing on a group outcome rather than an individualized outcome. So a good example of this is like if a person has cancer and they go in and they get screened, they're like, okay, let's say you have breast cancer, and they, do some of their starting testing. They say, this is the pathway we should follow. You do this chemo regimen, or you do this therapy, or you do this surgery regimen. And then what happens, though, is when we're looking at it through a precision medicine eye, we take a piece of that patient's cancer, we look for the genetic alterations in that cancer that make it tick and talk, and we attack those as well. So I really do think that when we look at patients, broad guidelines are, of course, always going to be helpful, but each person is unique. And I think that taking those unique aspects of each patient's journey into consideration does lead to better outcomes. It is a genetic underpinning. We understand what causes the disorder in my family. So understanding that means that we can target that alteration with some kind of treatment and address it directly. So for example, my family has a progranulin mutation in our family. We know that individuals that are lacking in progranulin have issues with neuronal cell health. And we know that we're missing about 50% of the normal amount of progranulin that we need. So in a targeted setting, you could say, oh, you have a mutation in this gene. You're missing 50% of the normal amount. Let's supplement it some way. That could be like a progranulin infusion or blocking its degradation, or even with gene therapy, which we're seeing very frequently nowadays. So personally, to me, that's how I think the targeted and precision medicine aspect comes into play. First and foremost is recognizing the fact that they both have a genetic underpinning, cancer and FTD. I do think that even though the genetic causation for all cases of FTD is not known, I think we are pretty confident that there is some kind of genetic underpinning for most FTDs. Cancers become so comfortable with that. People who research cancer, I think we need to be able to do that also in the realm of FTD. I think a great example of that is when I think about the way clinical trials are conducted for cancer versus the way they're conducted for FTD. A lot of times with FTD, we're looking at, okay, we are looking at a person with specific genetic, causation. We're treating towards that genetic causation, and we're seeing, does it slow or stop disease progression? But I think as we're seeing is that FTD is a lot more nuanced than just the gene causes it and then it happens. I think there's other things that happen at certain points in the disease process that we don't fully understand. And cancer is comfortable in that aspect because they do a lot of these basket trials where they're like, let's attack this cancer at all these different angles. They do combinations of treatments that make logical scientific sense that are backed with safety data. So a basket clinical trial is they say, okay, an individual has X, Y, or Z type of cancer, or they have X, Y, or Z type of mutation. And they say, here's a list of medications that can address either that particular type of cancer or that particular genetic mechanism. And they apply them in combinations to see, are there better outcomes when you mix and match? And that's something they do. And I think that, like I said, because we don't fully understand the mechanism of FTD, or they're complicated, we really have to look at it from different angles. And that's why I think we're seeing such an increase in treatment options for cancer, because they utilize that trial model data. And I'm hoping that we could see that in the FTD fight. I think a good example is not GRN, would be something like C9, right? Where we don't always understand, is C9orf72 caused just by the aberrant protein created, or is it that mixed with a lack of one copy of C9orf72? Is it a dual mechanism? I think we really need to be more comfortable attacking FTD from multiple angles, just like they do in cancer. There are plenty of perceived pros and cons when it comes to clinical trials. I always use the word perceived because because I think, right, it's a very arbitrary term and people can look at things with different weighted values. But I think overarchingly, when we think about what the pros of clinical trials are, is that you have treatments in either the absence of existing treatments or treatments that may be better and have less side effects than already available treatments. This is always a lesser, pushed pro, but the truth is that every time a person is involved in a clinical trial, they're advancing medical knowledge. And I think that is underrepresented as a pro, but it is a huge pro because we gather so much important data doing a clinical trial that we can take and map onto diseases and how to treat them better. And then I think the most important, and it's a commodity not to be wasted, is hope, right? I think particularly when we look at FTD, I think we're looking for that little beacon of hope that says that maybe our future is not going to be so grim as we perceive it to be. Ends up being in a clinical trial, right? Because there's the good, then there's the bad. As I think the first thing that comes to mind is the risk of being in the control group. In clinical trials, particularly those in phase 3, there's usually a group of individuals who are not receiving the treatment versus individuals who are receiving the treatment. Now, even people in the control group many times do end up having access to the drug at some point in the trial, but there is a period of time where they don't have access. So that could be seen as a con, and I think particularly in the setting of rare disease, that could be seen as a con if things are progressing. Another con, of course, is, you could be in this trial hoping and hoping and hoping for a positive outcome, and the drug fails. That's, I think, another thing that does happen that people underappreciate is that these drugs, while having great scientific rationale, don't always translate perfectly from the lab to human individuals. So I think that's another con. And I think, this one is kind of, thrown out there a lot, but people say it's like big time commitment. And it is a big time commitment. You usually have to go to several appointments. And depending upon what's being studied, you could have to get, MRI scans or blood draws or other, related tests. But, it's all about weighing out the risk and benefit in your own mind. So some people see the long time commitment as a con, and some people see it as just part of the game. So I'll tell you this. My mom did a gene therapy trial. So my mom actually wasn't in a control group. The disadvantage for my mom, which I think is a huge issue we see across the FTD spectrum, is that it took my mom several years to get an accurate diagnosis. And, with CureGRN-FTD, it is a fast-moving illness. So there was about a 3 to 4 year gap between my mom's symptom onset and when she received treatment. And, as I said, there seems to be some point in neurological disease progression where it goes from interventional to terminal velocity. And I think my mom, unfortunately, was at the point of terminal velocity when her treatment began. I will say the hope that it instilled in us, to hope that we were doing something, was so important. We wanted my mom to have that hope as well, and she did up to the very, very end. And I don't look at it as a failure at all. I think my mom did her part for science. I think we're doing our part for science. And I know, my mom, when she passed, she donated her brain as well. We are all hoping that everything that was put into that trial for ourselves and for my mom will translate, hopefully to my sister and I, God forbid we need it, but also to anybody else who has to face this kind of disorder. One other point I do think that's important to bring up, though, is, we do live in a day and an age where I think there are some ethical questions arising around utilizing symptomatic people as controls, particularly in the setting of rare disease. That's a question and maybe a talk and a whole other time. But I do think that with the advent of digital controls and large cohort data we have from other research studies, we could, maybe see a reduction in control trials in rare disease. For me, when I think about pharmaceutical companies and the inclusion of individuals who are at risk for FTD as well as those symptomatic, I think that there's been this weird push by pharmaceutical companies to create this odd third disease state where it's they call them pre-symptomatic. But I think the truth of the matter is that when you're a person who has a loss of function mutation in GRN, you are pretty much destined to get FTD or an FTD syndrome at some point in your life. I think you're pre-symptomatic from the day you're born to some extent. We know that most people develop the disorder in their 50s and or 60s, but that doesn't mean that it can't happen at any point in adulthood. I think that when they're designing clinical trials for this disorder, just like they've done with other lysosomal storage disorders. When you think about, what we currently believe that GRN-FTD is that it is a atypical lysosomal storage disorder, that I think we need to start treatment as soon as possible in our pre-symptomatic population to see if we can prevent the disease from occurring or at least push the date of symptom onset. And I think that they need to go hand in hand with the people who have symptoms. That's my personal opinion. I think there is good scientific rationale for it as well. I think we know in study lysosomal storage disorders that the longer it goes untreated, the more damage that occurs to the organs that are impacted, the worse the prognosis is. And I think that can directly translate to this disorder, because I think there has been some data released about certain trials or other, research studies that show that early intervention is where we can maybe stop the disease versus being in active disease state. As a person who works in genetics and does have a genetic risk to some extent, is that I don't think that this needs to define our lives. I think we live in a golden era of science. I think we need to trust science. I think we need to have access to science. I think, when I think about, particularly for clinical trials, when it comes to neurological disorders or rare disease, I think patients need to be given a little bit more deference. I think we have, when we talk about, if you're an individual who's into ethics, one of the key pillars of ethics is autonomy. And if we are people that are saying, I want to take the risk, I'm willing to take the risk, let us take the risk. Let us see what we can do. Because it's our right to have that hope for a healthy future and do whatever we need to do to mitigate our risk.